all, I want to thank the organization for this opportunity to share my work. I'm a PhD student, and I'm in my second year. I'm here to present some of the work that I'll be developing in these 2 years of PhD. So today I'm going to talk about the rhizobacteria, encapsulation, and volatile organic compounds, and their effects on plant growth under drought tolerance, or drought stress. As you all know, drought is expected to increase in severity and increase the periods of time, as increasing also in the climate change, and this can have different impacts on plants. They can lead to a reduction of photosynthesis rate, weaken their natural defenses, lead to a slow shoot development, also the accumulation of reacting oxygen species, and also reduce the nutrient uptake. The plants present some mechanisms that can help them to overcome these stresses. They can produce some compounds like flavonoids, polyphenols, and terpenes that will help to eliminate the ROS. Also, production of osmolytes that will help them to adjust their osmotic regulation under like for example, the salinity stress and drought stress, and also plants present some morphological adaptations like the root structure, reduced leaf area, and decrease in stomatal conductance. Also, the plants are not alone in the soil and in the environment. In the soil, they have like friends that are called the rhizobacteria, that are the bacteria that are present nearby the roots. And these bacteria can have a different type of mechanisms. They can produce like phytohormones, hormones, produce volatile organic compounds. They can lead to abiotic stress tolerance by the plants, and also increase nutrient availability in the soils. Like for example, the increase solubilize nitrogen, phosphate, and potassium, and also increase iron sequestration, production of ABA, gibberellins, and jasmonic acid. Also increase tolerance to different types of stresses like drought, salinity, and temperature. And also one of my main areas of study is the volatile organic compounds. Bacteria can release the different types of compounds that these volatiles present high volatility, low molecular weight, and high vapor pressure, so they can diffuse rapidly in the atmosphere. Also, they can be constitutive or induced by stress or some kind of stimuli. And also, they have high chemical diversity. Okay, I'm also always... okay. Like, for example, it's already been published that, for example, astuine, indole, and also 2-3-butanediol present some plant growth and health. Also, some of them present some antibacterial and antifungal activity, and also they are very good in the mediation of microbiology communities in the soil, for example. And due to their high chemical diversity, they are a huge amount of compounds that can be studied, and also we don't know how they can increase the plant tolerance or the effects on bacteria or in plants. So this is a huge gap in the science that is good to study. So one of the main problems of apply bacteria and apply volatile organic compounds is that the soil is presents some heterogeneity, and it presents some conditions that are adverse to the bacteria. And so one of the solutions that I am testing is encapsulation. What is this? Is to make a protective shell or a capsule around the active ingredient or the cells or bacteria. So there are different techniques like freeze drying, emulsion, ionic gelification, spray drying, but I'm using the extrusion for the bacteria it's more simple, and to apply in a larger scale is simple than the other ones, and more cheap also. There are numerous advantages like the increase in bacterial viability, also the maintenance of metabolic capability, also increase the storage and release time when applied to the soil, and also increase helps the bacteria to tolerate different stresses and adverse conditions in the soil. For that, they are already been applied in different areas, like for example in the agriculture, also in the food area, and also in the pharmaceutical area. So my main objective is to obtain biostimulants with microencapsulated volatile organic compounds and bacteria capable to increase plant drought tolerance. For that, I have several tasks, like, for example, the first one is to screen bacterial osmotolerance and plant growth promotion abilities. And second one is to, in this set of bacteria, to select some bacteria that can increase plant growth under drought stress. After that, I will select some strains and do some tests that I will see if these bacteria selected can increase plant growth under the volatile release. Also, after, I will proceed to the encapsulation, do test some techniques, and try to encapsulate bacteria and also volatile organic compounds. And for last, I will do encapsulation validation, some omics, and also the biochemical analysis to see if the applied bacteria and also the applied volatile organic compounds can increase the plant tolerance, and to see what is the differences in the plants and what are the mechanisms that the plant can trigger in the presence of these stimulants. For that, I want to test drought, so I need to find a drought tolerant bacteria. So I selected bacteria isolated from dry environments. Also, I tested for their osmotolerance and also for their PGPR traits. After that, from the around 100 isolates, I select 54 that showed higher osmotolerance and also good production of PGPR traits. These 54 isolates were selected for the test 2, that is the bacteria promoting plant growth and drought tolerance. For that, I selected maize as a plant model, and I subjected the maize to drought with a 35% of water holding capacity of the soil, and also I inoculated the 54 selected strains one by one also in a maize subjected to drought. I grow the plants in 2 weeks in greenhouse conditions, and I assess the root and shoot weight and length. With this, I selected 14 isolates that were the ones that showed the best ability to promote or root or shoot or both in both root and length. And these 14 isolates will be selected for the next trials. So the next trial is the bacterial volatilomics. So just to see if the bacteria can increase plant growth under, in this case I will do in a Petri plate with 2 sides with a division. So the contact between the plant and bacteria is only by the atmosphere, so is not a direct contact. And I selected the mannitol for the osmotic stress in the plants, and to subject the bacteria to osmotic stress, I increased the agar concentration in the medium just to increase the osmotic pressure in that part of the media. Okay. After that, I selected only 4 isolates. I used not only this approach, but also in a closed flask approach with maize. So as you can see, I selected the D12, C18, O220, and F11. The D12 here we used lettuce, but in maize it was the one that showed a higher induced tolerance and growth compared to the other ones. Here I also selected the C18 and O220, and the F11. It's like a negative control. That is the one that showed a little variation compared to the control. It will be the ones that I will be tested to see if the volatiles released by the bacteria can be different and if have some in common. For that, I will use the approach that Paul already explained, GCMES analysis, to try to identify some volatiles. I don't have the results yet, but I made I tweeted a little bit today, and I saw that between the control and the osmotic stress of the same bacteria, the clusters are different. That means that on the control and osmotic stress, the volatiles released by the bacteria are different. And I already saw that some of them present some capability of plant growth compared to the bibliography. So for the next, I tried the encapsulation. I will do the extrusion. So I will use alginate. It's a cheap one. It's one of most cheapest agents for encapsulation, and also it's biodegradable and they have a lot of advantages. And also to complement the matrix, I will supplement the alginate with starch, like an energy source for the bacteria when they are encapsulated. So also after the encapsulation, I checked for the structure and morphology of the capsule, the bacterial survival, and the bacterial release first. And for the capsule structure, we can see that the first one is only alginate, is the surface, and we can see some cracks. Is due to the fragility of alginate. But with the supplement of starch, the starch filled the matrix inside of the alginate and increased lead to a reduction of the fissures in the surface and also enhancing the spherical geometry of the capsule. Also, this is the inside of the capsule, only with alginate. Due to the porosity of the capsule, when I freeze-dry the capsule for the same analysis, the structure collapsed. So we can see the detailed porosity of the capsule, but in a closer look, we can see the little lines of the porosity. But with the supplement of starch, the starch filled the pores of the alginate and increased the resistance, and also we can see the uniform distribution of the starch in the surface and also in the inside of the capsule. Okay. So for next, I saw the bacteria survival after 1 month, and I can see that almost bacteria tested showed good results, with one of the 2 or 3 lead to decrease in like true logs by the 20th day of experiment, but in majority of the strains tested, they show the good results of bacterial survival. After the bacterial release, I put the capsules inside of PBS, and I took a liquid in different times at also 1 month, more or less. As I can see that almost at the 8th day, the maximum bacterial release was the highest. With some strains or another one that showed a more prolonged release over the time, is this one. But the majority of them released the maximum growth in this one. Okay, I still trying to perform this. I'm trying to encapsulate some volatiles. I already tested some strategies, and this one was the most promising one. So I prepared an emulsion. Also, I added the beta-cyclodextrin because I want to retain the volatiles inside of the capsule for long periods of time. And also, when if I want to make this a product, I need to retain this capsule, the VOX, the majority of time that I can. So I added cyclodextrin, volatile, and I performed the emulsion. And after, in the emulsion, it was alginate dissolved. And I also made the capsules by extrusion. So I encapsulated the emulsion. So what I supposed to get is like a mini capsules inside of a giant capsule. So over time, the giant capsule will start to degrade in the soil and start to release over the time the volatiles that are inside of the capsule. This I'm still trying to find the best formulas and the best concentration for volatiles. I already tested one of them, but I think I put too much volatile, and the result was not what I expected. So for the last, I already tried to encapsulate the bacteria, and already tested the effects of the bacteria inside of the capsule. I took the same approach as before, but instead of direct inoculation, I applied the capsules beside of the seed. And also I took the same approach, and as I can see is that I have some differences between the strains, when some of them showed good results, like the F43, D8 and F3. As I can see in the shoot length and shoot weight, the F3 showed good results, Like it's not, it's like increasing 5%. It can be not too much, but it's a good value. These values are compared to the drought control. So the F3 is the one that shows best results to the shoot, and in the root, the E7 and also the F3 show the good results also. So I performed the biochemical analysis. I didn't put here because I think I'm taking too much time. But the major difference that I saw is the increase in the sugars content and also in the proline content compared to the drought control. And also I saw the increase in chlorophyll content, show the high metabolic capacity by the parameter of electron transport system. I saw an increase in metabolic capacity compared to the other ones when not exposed to the capsules. Okay. And it's this for now. So thank you for your attention. If you have any doubts, just ask me.
Thank you very much. That was really interesting, and you've really done a lot, and it's really exciting for the future. Does anyone have any questions? I have a question. I didn't see that.
Thank you for presenting. I was wondering about the particle coating with starch.
Yes.
I just saw that couldn't be or like not so good aspect for like fungal contamination, because we know that.
Yes, fungal.
In terms of stability.
Yes, I saw that. I performed the trial like 4 or 5 times, and I sterilized the reagents very well, but in the first time I didn't sterilize it well, and the contamination was bad. I had really fungus, but bad fungus. The contamination was horrible, but I use the starch because it's a natural polysaccharide. Yes, I can. Yes, I put it on the fridge. I don't have any contamination, but try to. I didn't lyophilize the bacteria, so I don't have that. So I hope that when I lyophilize the bacteria with the starch and encapsulated, I can prevent the contamination and store it for longer periods of time. But I didn't test that yet, so in the next trials. Thank you.
Any other questions? My question also got answered through this. So thank you very much.
You.
Thank you.